Let's travel back 61 years ago, when Sir John B. Gordon realized that he had reprogrammed the nucleus of a frog's skin cell by injecting it into an enucleated frog egg. It was found that it wasn't the nucleus that determines what a cell does, but rather the contents of a cell's cytosol that are able to dictate which genes are transcribed in the nucleus. Therefore, despite having the nucleus of a somatic skin cell, the enucleated egg cell behaved just like a normal frog egg. Back then, the presence of transcription factors were unheard of. They are the key regulators to cellular gene expression. It was not only 45 years later did Shinya Yamanaka identify the key transcription factors that dictated cellular reprogramming. When delivered to human dermal fibroblasts through retroviruses, they are able to induce cellular reprogramming through dedifferentiation, a process where a once unipotent cell figuratively travels back in time and becomes pluripotent once again. These pluripotent cells are able to differentiate into various cell types. Here we have figures from his findings, where you can see that OSKM-treated HDFs resemble embryonic stem cells and are able to spontaneously differentiate. However, Yamanaka's stem cells were not ideal due to the use of viral vectors, which may lead to insertional mutagenesis and unanticipated genetic alterations on elements that play a crucial role in maintaining cellular proliferation. These genetic changes may increase the risk of tumorigenesis, limiting their safety and clinical applicability. It was not until studies around 2010 successfully eliminated transcription factors in inducing pluripotency in somatic cells. By this time, researchers were able to replace most of the Yamanaka factors with small molecules to induce pluripotency in mice. These stem cells were called chemically induced pluripotent stem cells, the main focus of this video. These small molecules target epigenetic and signaling pathways to alter gene expression without the use of transcription factors. The main concept behind CiPSCs isn't that complicated. We need to find a combination of drugs that are able to activate the pluripotency gene network and suppress mechanisms that reinforce specialization and differentiation. This aims to push the somatic cell into an intermediate plastic state which resembles natural dedifferentiation that takes place during limb or tail regeneration in axolotls and salamanders. By 2013, mice somatic fibroblasts were successfully reprogrammed using a cocktail of small molecules. SKM was successfully replaced by Repsox and Parnate. OCT4 was tougher to replace, requiring a lot more molecules. So how exactly do these small molecules work to replace OSKM? Epigenetic modifications maintain a differentiated state and is a major barrier to reprogramming. Histone deacetylases remove acetyl groups from lysine residues on histones to allow a tighter and more condensed wrapping of DNA. Additionally, they ensure the chromatin suppression of pluripotency genes in somatic cells. DPA functions as an histone deacetylase inhibitor, allowing accessibility in said regions. On the other hand, the trimethylation of lysine 4 is generally associated with active transcription, whereas the removal of these methyl groups can repress gene expression. Parnate functions as an H3K4 demethylation inhibitor to allow active transcription of these pluripotency-related genes. DZNEP is also an indirect methylation inhibitor. Aside from epigenetic regulators, signaling pathways like GSK3-beta and TGF-beta may be two major barriers of reprogramming. CHIR99021 inhibits GSK3-beta in the Wnt pathway, upregulating STAT3 transcription. 616452 inhibits TGF-beta signaling through ALK5, which prevents inflammation-driven fibrosis, which is known to block dedifferentiation during regeneration. Last but not least, OCT4 was successfully replaced by increasing cellular concentrations of CAMP through forskolin that directly activates adenylyl cyclase. This activates the exchange protein directly activated by CAMP signaling pathway that contributes to downstream epithelial gene expression while decreasing mesenchymal gene expression through its downstream effector RAP1. Murine embryonic fibroblasts treated with forskolin, GSK3-beta, and MEK inhibitors expressed epithelial markers EPCAM and CDH1. This cocktail is not the only combination of chemicals able to induce pluripotency in mice somatic cells, as various other combinations of small molecules that target the key pathways mentioned beforehand are able to produce similar results. However, chemical reprogramming of human somatic cells to pluripotent stem cells was only achieved recently in 2022 with the cocktail listed on screen. Human somatic cells perform specialized functions, giving them a higher degree of epigenetic stability and reduced plasticity compared to mice. Hence, more specific epigenetic and signaling inhibitors needed to be added to the mix. A key barrier to chemical reprogramming in human somatic cells is the JNK pathway, which is mainly involved in stress and inflammation responses, which blocks the dedifferentiation in reprogramming. Hence, inhibiting this pathway was crucial for generating CiPSCs. In the present day, groundbreaking studies showcased the potential of CiPSCs in treating various conditions. One study demonstrated the successful transplantation of iPSC-derived islets, thus resulting in the first cure of type 1 diabetes. Another study focused on iPSC-derived corneal epithelium for transplant surgery, marking the first successful vision restoration treatment. These advancements highlight the therapeutic applications of CiPSCs, offering hope for patients with diabetes, vision impairments, and other diseases, pushing the boundaries of regenerative medicine and personalized therapies. Here's a recap of what we learned. These are the targets of small molecules used for chemical reprogramming. Aside from transcription factors, signaling receptors, and transduction proteins, along with epigenetic modifiers, are key regulators of gene expression. Targeting them will have an impact on cell fate. The future of medicine is exciting, and as this field continues to evolve, we're only beginning to understand its true potential. Thanks for watching, and stay tuned for more insights into the cutting-edge science shaping our world.